Hello students, I am your science teacher Preeti ma'am and I am going to teach you chapter 2 microorganism of standard 8. The topics that I am going to cover in this chapter are food prevention, different methods in which we are going to stop the food from spoiling. And the methods that we use it are the dehydration process, heating process, refrigeration and freezing, vacuum packing, canning and adding preservatives. And with that, I will be teaching you about the nitrogen cycle also. Food preservatives, or how to prevent the food from getting destroyed, from getting spoiled. A variety of methods are followed to stop food from spoiling. The first method is the dehydration, means removes moisture from the food. Dehydration is one of the oldest methods of food preservation. It removes the moisture from the food and so it stops the microbes from growing on it. Dehydration also decreases the weight of the food, so it becomes easy to store and to transport. But when we use the dehydration process, some of the vitamins like vitamin B complex and vitamin C, they are destroyed when the food is dehydrated. Mushrooms, most of the fruits and all, they are preserved using the dehydration method. The next food preservating process is the heating. Heating food kills most of the microbes, most of the bacteria, most of the microorganisms. So that is why water and milk, they are heated till they are boiled and before we drink them. So heating before drinking. Louis Pasteur, one of the famous scientists, found that heating food to around 60 degrees Celsius would kill the microbes and bacteria. So milk is heated to around 72 degrees Celsius for about only 15 seconds and it is rapidly cooled to about 4 degrees Celsius. The high temperature of 72 degrees Celsius kills the bacteria in the milk and the rapid cooling of around 4 degrees Celsius prevents the recontamination, and this method is called as pasteurization. The heating and rapidly cooling of milk. Refrigeration and freezing is the next food preservation method. Most of the microbes need optimum temperature to grow and to multiply. Cooling food makes them inactive and also it slows down the growth and their multiplication also. Food does not spoil for a few days if it is stored at around 5 degrees Celsius for some months and at 18 degrees Celsius. When the food is thawed, thawed means to make it hot, warm and soft, the microbes become active again and the food has to be used very soon, otherwise it gets destroyed. And meat, this is meat, and meat is preserved by the process of refrigeration and freezing. Next topic that we are going to discuss is the vacuum packing method of food preservation. You know students, many microbes are aerobic. Aerobic means they use oxygen for respiration. When food is stored in vacuum, the microbes cannot grow means it stops growth and multiplication due to the lack of oxygen. Means if there is lack of oxygen, if there is less oxygen or no oxygen at all, the microbes cannot grow. This method is commonly used to store the nuts, the grains, coffees and many other things. Some of the chips will get crushed in the vacuum packing bags. The air inside replaced with the nitrogen. So vacuum packed bags contain nitrogen in them. The next preservative method is the canning process. Students, I have already told you that microbes need air, oxygen to grow and to multiply. Microbes need air, and so storing food in airtight cans, it helps it to stay unspoiled for a long time. The food does not get spoiled when there is lack of air or less air. So we have to store them in airtight cans. And the foods that we store in such cans are the jams, the fruits, vegetables, fishes. They are canned. The best example of food preservation is adding preservatives. You know, students, some edibles, some food items that we eat, edible substances either kills microbes or prevents their growth. So oil, vinegar, they are the examples of substance that prevent microbial growth. So students, food materials are pickled in oil. You might have seen that. And vinegar to store them for a long time. And such things that we store are the vegetables, fish, fruits and the meat. They can be preserved in this way. So what to do? Salt. Salting is mainly used to preserve the meat and the fishes. Some vegetables like chilies and mangoes, they are also preserved by adding salt. So salt forces the moisture out of the food. It removes the moisture from the food. It removes the water molecules from the food and the microbes also. And thus it prevents the microbes from growing. Another preservative method of adding preservatives is the sugar. It is used to preserve the fruits. You might have seen the jams and jellies. They are mostly preserved by adding sugar in them. Chemical preservatives also help us a lot. Chemical preservatives like sodium benzoate, sodium metabisulfate, they are used in commercial purposes in the preparation of pickles, different types of squashes, the ketchups, the jams, so that the food is stored for longer time. The fish that is stored by the addition of the salts. This is the salted fish. Students, this is the most important topic, the nitrogen cycle. This is so important that it is added to your syllabus even in the 9th standard. Nitrogen is the major, most important constituent of the Earth's atmosphere. It is a part of proteins and DNA. And you know, students, this DNA, it is the hereditary material in the cells. But some plants or most of the plants cannot use nitrogen as it is. It cannot absorb the nitrogen. It has to be converted to other forms before the plants can take it up. So, the process by which atmospheric nitrogen is converted to usable forms of nitrogen, it is called as nitrogen fixation. The atmospheric nitrogen fixation means it is a process by which atmospheric nitrogen, the nitrogen which is present in the atmosphere, which the plants cannot absorb it because plants need the nitrogen to prepare the food. And the plants cannot use it by itself. It has to be converted into that form in which the plants can use it, and this is called as the nitrogen fixation. Atmospheric nitrogen fixation. The large amount of energy in lightening oxides a small amount of atmospheric nitrogen into its oxides. Means nitrogen, by the process of lightning which happens in the rainy season, it converts nitrogen into its oxides. The nitrogen oxides, it dissolves in the rain to form dilute nitric acid. And the nitric acid, it reacts with the compounds in the soil, forming soluble nitrates, and that can be taken up by the plants. So here this process is explained over here. The nitrogen which is present in the atmosphere, due to lightning, it reacts with the oxygen and forms oxide. This nitrogen oxide, it reacts with the oxygen which is again present in the atmosphere and converts into nitrogen dioxide. And this nitrogen dioxide, when there is rainfall, it mixes with the water and forms the dilute nitric acid, and this dilute nitric acid can be in the absorbable form by the plants. It can be absorbed by the plants. Biological nitrogen fixation. Students, we have already learnt about the rhizobium bacteria which is present in the leguminous plants of the peas and the beans and most of the other leguminous plant. And this we have already studied in chapter number 1. This rhizobium bacteria, it fixes the atmospheric nitrogen. Other nitrogen fixing bacteria and blue-green algae, they also fix the nitrogen directly in the soil. Nitrogen assimilation. Plants take up and use the nitrogen compounds because I told you that plants cannot use the nitrogen directly. So it has to be converted into the form that can be absorbed by the plants. And that nitrogen compounds are used by the plants and formed as a result of nitrogen fixation through their roots and make proteins for their growth. The nitrogen becomes a part of plant tissues now. The nitrogen is passed on to herbivores and to carnivores, and so on. It passes in the food web, the food cycle, through the food chain, and becomes a part of the animal tissues also. This conversion of nitrogen compounds into tissues, it is called as nitrogen assimilation. Because students, you know that plants are only the autotrophs. Rest all they are heterotrophs, like we discussed in the last chapter. Heterotrophs, they cannot prepare the food. They are completely dependent on the plants. So these proteins which are made by the plants, when the animals eat it, then the food is stored in them also. Then proteins are stored in their tissues also. So this conversion of nitrogen compounds into tissues, it is called as nitrogen assimilation. This is the ammonification process. You know that when plants and animals die, microbes decompose the dead material. You know that there are many microbes which convert the dead materials. Ammonia is produced during this process. The process of formation of ammonia by fungi and bacteria when they decompose dead and waste material, it is called as ammonification. The process of formation of ammonia by fungi and bacteria, when the fungi and the bacteria, they decompose the dead and waste material, this process is called as ammonification. After ammonification comes the nitrification process. Some of the ammonia formed is taken up directly by the plants. The ammonia which is formed by the process of ammonification is used by the plants directly, by some of the plants directly, while the rest is converted into nitrates. And who converts these, students? The nitrifying bacteria. The nitrifying bacteria convert this ammonia into nitrates. So the process of converting ammonia in the soil is called as nitrification. The process of converting ammonia to nitrates in the soil is called as nitrification. And some of the nitrates are absorbed by the plants also. And next process, or the last process of the nitrogen cycle, is the denitrification process. Apart from the nitrates in the soil, is converted into gaseous nitrogen also, and that gaseous nitrogen, it escapes into the air, it goes into the air. The bacteria that are involved in this process, they are called as the denitrifying bacteria. And the process of conversion of nitrates to atmospheric nitrogen is called as denitrification. And this is the whole process of the nitrogen cycle that goes on. The process of the atmospheric nitrogen fixation means converting of nitrogen through the lightning, conversion into the nitric acids. Then biological nitrogen fixation goes on. And then we have the nitrogen assimilation. And then also ammonification, nitrification. And the last is the denitrification. Thus, students, we come to the end of the chapter number 2, and these are some of the questions that are attached to you that you are going to write in your notebooks. Let us discuss some of the questions. A substance that is produced by a microbe and kills other microbes, what is it called? Oil helps to preserve food by what? What are preservatives? That you are going to answer. Write the definition of preservatives and why are they added to food, for what purposes, and you are going to name any 2 chemical preservatives. You are also going to write the difference between the nitrifying and the denitrifying bacteria. And the 5th question, you are going to list the ways in which food can be preserved. There are different methods in which the food can be preserved, and you are going to write them all. And you are going to write the different steps of the nitrogen cycle. So this will be your task of the day. So bye-bye for then. Bye, students.